Hello students, welcome back to our channel. So today's class is on meiosis, a kind of difficult topic for students, but I'll make it as much as possible easy for you to understand. Okay, so let us get into the class. So meiosis, now before getting into the definition and proper class, let us understand what happens before meiosis I. Okay, so what happens before meiosis I begins? So yesterday I told you we have meiosis I and meiosis II. So today we will see what is meiosis I. Okay, so what happens before meiosis I? So there is an stage called as interphase. Okay, so interphase stage happens. So we have seen in mitosis that during interphase stage there is a phase called as S phase, isn't it? So in S phase what happens? There is the synthesis of the chromosome. So meiosis starts, so DNA replication occurs during this S phase. So before S phase, now before S phase, if there is 46 number of chromosomes, okay, after S phase what happens? After S phase, the same 46 number of chromosomes, but then 92 chromatids. So DNA molecule is being doubled. Okay, so this happens in the meiosis, before the start of meiosis I, that is during the interphase stage. Clear? Okay, so now let us go into meiosis I. Meiosis I. Okay, so meiosis I actually has 4 stages. Okay, prophase I, metaphase I, anaphase I and telophase I. Okay, so these are the 4 stages of meiosis. Okay, now this prophase in turn is said to be the longest phase. Okay, this prophase is said to be the longest phase and very important phase and very long phase. Okay, so you can say the star of meiosis is prophase. So prophase in turn is divided into 4 different stages. Okay, so what are they? Leptotene, zygotene, Pachytene, okay, followed by Diplotene, and then the last one is Diakinesis. So these are the 5 stages of prophase I. Leptotene, zygotene, Pachytene, Diplotene and Diakinesis. A easy trick to remember is LZPDD. LZP and DD. Just by heart this thing and you don't have to mug up the whole sequence. Leptotene, zygotene, Pachytene, Diplotene and Diakinesis. Okay, so we will get into this particular phase, very important phase in meiosis I, okay, one by one. Clear students? Don't get confused, just follow the class, you will be very much clear after this class. Okay, so we will go into leptotene first. Leptotene stage. So what happens in leptotene stage? So during leptotene stage what happens is chromosome first becomes visible as a thin thread like structure. Okay, so chromosome becomes visible as a thin thread like structure. So this happens in leptotene stage. Okay, so if I draw the cells of leptotene, you will see something like this. Okay, thin thread like structure of your leptotene. Okay, so why I am drawing the two different colors? Because now this is a meiosis process where a sperm and the egg has fused and they have two different chromosomes from different species, different individual, father and mother. Remember this, students. Okay, always remember this fact when you are studying about meiosis. Okay, so that's the confusion which actually is created in the student's mind, because you forget the fact that it is a meiosis which is happening between the chromosome here is from two different individual. Okay, so next stage we will see the zygotene. Clear? So you are clear with what is leptotene. Very simple thread like structure seems to happen. Now in zygotene what happens? Homologous chromosome come together. Homologous chromosome come together and pair. Okay, now what do you mean by a homologous chromosome? It is nothing but the chromosome I just told you, we get a chromosome from a mother and a father. So homologous means the same kind of chromosome, one each from a father and mother, they come together. Okay, same kind of a chromosome from father and mother. For example, a father chromosome having a trait of a eye color and the mother's chromosome having a trait of a eye color. For example, mother's eye color and father's eye color chromosome, they come together and they pair, and this is why we call it as a chromosome, homologous chromosome, because they carry the same trait. Okay, so imagine two books published in two different cities. For example, one is published in Chennai and the other is published in Delhi. Okay, there will be a slight misprint in the spelling, for example. Okay, so this misprint in the spelling is the eye color. The eye color of father is blue, the eye color of a mother is black. Okay, so this is the slight misprint, but then the trait is the same. The trait is the same, only there is an error in the spelling mistake. Okay, so this is what we call it as a homologous chromosome. Hope it's clear. Okay, so what happens in zygotene? The homologous chromosome, they come close and they pair. Okay, and the pairing of homologous chromosome is called as synapsis. The pairing is called as synapsis. Synapsis is nothing but pairing of homologous chromosomes. Pairing of homologous chromosomes is called as synapsis. Okay, and here I would like to introduce you with another term which is bivalent. Bivalent is nothing but a pair of homologous chromosome. A pair of homologous chromosome. Okay, a pair of homologous chromosome. Clear, students? Okay, so let us try and draw the diagram here. Okay, of a leptotene. So here I'll first draw and then I'll explain you. Okay, so this is I'll take blue color. Okay, so this is the two sets of chromosome which have come together, okay, and paired. So this is one pair and this is second pair. This is the nuclear membrane and this is the cell membrane. Okay, one pair and second pair. So this is the bivalent. This is bivalent, pairing of homologous chromosome together. This is one chromosome, this is second chromosome, and each chromosome has two different chromatids. Okay, so one from the father, one from the mother, one from the father, one from the mother. Okay, so homologous chromosome has come together and the stage is zygotene. Okay, clear? Right? Now next stage is, what is the next stage? The next stage after L, Z is P, that is Pachytene. Pachytene. Okay. So what happens in Pachytene? So in Pachytene, the chromosome becomes more thick and short. Okay, chromosomes becomes more thick and short, and homologous chromosome okay, exchange genetic material, that is what is happening here. Now here we will introduce the term called as crossing over. Exchange of genetic material, exchange of chromosome segments. Crossing over is nothing but exchange of chromosome segments. Exchange of chromosome segment is called as crossing over. Now remember students, so there is the exchange of chromosome segments, okay, but the segment is a non-sister chromatids. So exchange happens between non-sister chromatids. Don't get confused at all. I will explain through the diagram. You will be very easily understanding what I am going to say. Okay. So let us try and draw the diagram of a Pachytene stage. So here homologous chromosome, okay, the father's chromosome, the mother's chromosome, okay. I'll tell you what is actually happening and what is the statement I was telling you. Okay, so what has happened? Now here see what has happened. Now this is crossing over. So this is the crossing over. Okay, now before that I'll complete the diagram. The nucleus and the nuclear membrane. Okay, so here you can see the crossing over has happened between the homologous non-sister chromatids. Now this is sister chromatid, this is sister chromatid, but exchange is happening between these two. Exchange is happening between these two. Please zoom in when you are listening to this class, students, so that you are very clear with the concept. Now this too is a non-sister chromatid. So exchange is happening between a non-sister chromatid, and this is what I meant with the definition. Exchange of chromosomal segment, but then the exchange is happening between non-sister chromatids. Okay, so what is this stage? Pachytene. Okay, this is the stage is Pachytene. Clear? Okay, so this is what is happening in a Pachytene stage of a meiosis prophase I. Now we will enter into meiosis prophase I. The next stage is Diplotene. The next stage is Diplotene. Okay, so what happens in Diplotene? In Diplotene, two homologous chromosome begins to separate. Two homologous chromosomes begin to separate. Okay, they begin to separate, but they remain attached at certain point. Okay, but still they remain attached at certain point. So okay, I'll write it, but remain attached, at certain point. Okay, so this point is what you call it as chiasmata. Okay, this point is called as chiasmata. Chiasmata is nothing but visible points of crossing over. Visible points of crossing over is chiasmata. Okay, visible points of crossing over is chiasmata. Okay, so I will try and draw the diagram for you students to make it more understandable. So here what we have seen according to the explanation, so here, so this is the Diplotene stage, the nuclear membrane followed by the cell membrane. Okay, so according to this definition, two homologous chromosomes begin to separate. So in the previous stage what we have seen, in the previous stage we have seen the structure like this, crossing over has happened. Okay, so here you can see there is a separation being happening. Okay, the separation is started but still attached at one point, still they are attached at one point. Okay, and visible point of crossing over you will see. Okay, and that position is called as chiasmata. I will make it more clear in the next diagram what is chiasmata. Okay, so diplotene, what is this stage? This is your diplotene. This is your diplotene. Clear students? Okay, so what is the next stage? The next stage is diakinesis. Okay, so what happens in diakinesis? So as we have seen in diplotene, we have seen the stages of chiasmata being formed, right, visible. So here in diakinesis, final stage of prophase you can say, and here you will see the nuclear membrane, NMS, nuclear membrane disappears. Okay, nucleolus disappears and spindle fiber forms, begin to form. Okay, so ultimately what is happening? Cell is preparing itself for the chromosome movement to happen. Okay, cell is preparing itself for the chromosome movement to happen. Now here we will see what is happening in diplotene in diagram. So I will draw and tell you what has actually happened with your chromosome now. Okay, so this is what has happened actually with your chromosome. Okay, so can you understand now what has happened with your chromosome? So what has happened? In diakinesis, what is happening? The nuclear membrane has to disappear. So I am drawing it in a dotted form. Okay, nucleolus has disappeared already. So nuclear membrane, this is cell membrane. Nuclear membrane has to disappear. So here you can see the exchange of chromosome has already happened, and this point of crossing over, this is the point of crossing over which is chiasmata, which we saw in diplotene stage. Now this is what is called as chiasmata. Okay, clear? Okay, chiasmata, visible point of crossing over, chiasmata will be started to be visible during this stage. Okay, so this is what stage? Diakinesis. So diakinesis is nothing but cell is preparing itself to undergo the next stage, that is enter into metaphase. So prophase is over. So what has happened in prophase? Prophase, what has happened? The exchange of genetic material between the homologous chromosome has happened. Okay, homologous chromosome has happened, and this is why we are not exactly the same as our father and mother, because there is a shuffling of genes which has happened. Clear, students? So, here diakinesis has happened. Okay, so now in metaphase, now next stage, now prophase is over. Now after prophase, what is happening? The prophase, after prophase, as usual, metaphase. We will label it as metaphase I because we are talking about meiosis I. Okay, so in metaphase one, what happens? The stage, the event which you will see is homologous chromosome pair arrangement happens at the equatorial plate. Equatorial plate. The chromosome is arranged at the equatorial plate. I am not writing it in detail, instead I will draw and show you. Okay, so the chromosome arrangement happens at the equatorial plate. Okay, so here is your chromosome, homologous chromosome, which pair together, which come together, okay, and align in the equatorial region. I will draw in this corner so that it is more clear to you all. Okay, so this is the point of exchange which has happened. Clear? So this is homologous chromosome 1, homologous chromosome number 2, which has come together. The nuclear membrane is already disappeared. The spindle formation has happened already in diakinesis, isn't it? So I forgot to draw here the spindle fiber formation. Okay, so here also after the homologous chromosome they pair with each other, the spindle fiber formation happens, and this spindle fiber attaches to the centromere, as you all know, right? So this is one pair of chromosome, homologous chromosome, this is another pair of homologous chromosome. They align at the equator. Okay, so this is the metaphase 1. So what happens is the next stage is anaphase I. Anaphase I. So anaphase I, all of you know, this is where the actual reduction happens. Okay, so what happens is the homologous chromosome, they separate. Okay, the homologous chromosome, they separate. I'll draw and tell you instead of explaining it through the writing portion. Okay, because that will make you more clear. Anaphase I, the chromosome. Okay, so here the another set of chromosome which has gone. This is the next set of chromosome, and the pairing has happened here, the pairing has happened here, sorry, the crossing over, and the crossing over happened here, the crossing over happened here. Okay, so here what is happening now? Students, concentrate. I want you to concentrate here a bit so that you will understand what actually is reduction. So here you can see a homologous chromosome. Okay, they separate the homologous. Now here we saw this was one set of homologous chromosome, isn't it? Yes. So now here you can see these are getting separated. This is moving towards that end, this direction, and this is moving towards this direction. Clear? So homologous chromosomes are separating. So half of the chromosome is going there, half of the chromosome is coming here. Okay. So now what is happening actually to your chromosome? This 2n, which was here actually in a 2n stage, now is becoming n and n. Are you getting my point? The 2n, which was homologous chromosome, father and mother, is now after genetic shuffling getting separated into n and n. Okay? So, Anaphase I, the chromosomes are getting separated. Okay? One chromosome of each pair goes to the opposite direction. Remember students, the centromere is not splitting in Anaphase I of meiosis. The centromere is not splitting, which you saw in mitosis, and this is the reason why the 2n becomes n. Here the 2n chromosome now becomes n. Clear? Now this is 2n. Okay, this is 2n. Now this becomes separated to become n. Getting it? Okay. So now again through diagram only, because I will not write much. The last stage is telophase I. So what happens in telophase I? The chromosome reach to the opposite pole. So again I will draw instead of writing much, so that you understand, because I am speaking as well, so that you get my point. Okay. So chromosome reach to the end. Okay. Chromosome reach to the end because of the spindle fibers. Okay. So this happens in what stage? In telophase I, right? So here the exchange of chromatids had happened already. So this is the point of chiasma. This is the point of chiasma. Okay. And what is happening in telophase? The next stage, what is happening in telophase is reappearance of nuclear membrane. Reappearance of nuclear membrane. Okay. The cell is one, but then there is reappearance of nuclear membrane after it reaches to the opposite pole. Okay. So telophase I causes the movement of DNA to the opposite pole and reappearance of the nuclear membrane. So after this, the stage which is followed is, cytokinesis. Cytokinesis, as all of you know what is cytokinesis, it is nothing but the division of cytoplasm. So cytoplasm division forms. So what will happen? You will see two nucleus has formed already. Okay, receiving two sets of homologous chromosome. Okay, two sets of homologous chromosome with genetic material exchanged. Okay, with genetic material exchange, and now this has become n number of chromosome instead of 2n. Okay, because now the father and mother chromosome has separated after the genetic shuffling. Okay, and now this cell will now deepen, the cleavage furrow will deepen as we have seen in the previous classes, and give rise to two different daughter cells. Okay, two different daughter cells like this. So now you will get two different daughter cell with two different nucleus, and the nuclear DNA will be made up of n number of chromosome. So that's the result of meiosis I. Okay, so that's the result of meiosis I. Clear students? Hope it's clear. Yes. So meiosis I will give you two telophase, diploid cell becomes haploid, still having two chromatids. Clear? Hope the class is clear, and I will see you in the next class. So in meiosis I we have seen the cell gives you two haploid daughter cells. Okay, so after the division your result is two haploid daughter cells. So tomorrow I will tell you in detail about meiosis II. what happens in meiosis II. Okay, so hope the class is clear. Any doubts, do comment me and let me know. Please do like and share my classes, students, because I really get motivated when you do so. Please do that.